Imagine you're a new mom holding your beautiful newborn for the first time. But just minutes after his birth, the doctors notice something concerning and order a series of tests. All of a sudden, your joy is met with fear and uncertainty. The doctors come back with some concerning news. Your baby's ammonia levels are dangerously high. And as they dig deeper, they notice something quite unusual. Just one letter out of place in his DNA has created a faulty enzyme that stops his body from safely breaking down protein. Without that enzyme, toxic ammonia levels build up in his body, threatening his brain, his liver, and his life. For now, the only treatment is a liver transplant, but that requires your baby to be older and for there to be an organ donor. The longer you wait, the sicker your baby will get. But what if we didn't have to wait? What if we could precisely correct that single error in his DNA? What if instead of just treating symptoms, we could cure diseases on demand? Over the next several months, scientists race against time to develop a personalized CRISPR medicine designed to correct the exact mutation in your baby's DNA to restore the functional enzyme and stop the deadly metabolic disorder. This involves testing and manufacturing the medicine at unprecedented speed to ensure that it is safe and works in time to manage your baby's condition. Eventually, scientists think that they found a solution. Your doctors tell you, "We believe we've developed a personalized CRISPR medicine designed to work precisely for your baby, and we believe this will address the root cause of his disease." But it's never been tested on anyone before. All of a sudden, you have the gift of time. You would do anything for your kid and decide that the best possible scenario is to try the custom gene editing therapy. So at just 6 months, your baby receives the first infusion. By 10 months old, your baby is thriving. He's meeting his developmental milestones, growing stronger every day, and finally, he's able to go home. This is the story of baby KJ, who just earlier this year received the world's first personalized CRISPR medicine, developed by teams across multiple institutions led by Dr. Kyun Musunuru and Dr. Rebecca Ahrens-Nicholas. And I'm so happy to share that just earlier this week, baby KJ was finally able to go home after 10 long months in the hospital. But his journey is more than just a medical milestone. It's a beacon of hope for the millions of patients and families worldwide living with rare and common genetic diseases. Over the last several decades, scientists have made incredible progress understanding how our genes influence our health. Think of our DNA like a massive instruction manual, billions of letters that describe how our bodies grow, function, and heal. But sometimes there's a single mistake in that manual, and that might cause something to go wrong. As we just heard, just a single typo can cause deadly disease. And so scientists have dreamed of finding a way to fix these typos without changing the overall story. In 2012, my former PhD advisor, Dr. Jennifer Doudna, and her colleague, Dr. Emmanuel Charpentier, won the Nobel Prize for their work discovering CRISPR technology and showing that it could work like a programmable editor, paving the way for CRISPR to repair the genetic letters. So how does the CRISPR technology work? There are two main components. The first is a CRISPR protein, here in yellow, and a guide RNA in green. Together they form a complex. Like a GPS, the guide RNA can be reprogrammed to target any DNA sequence, enabling the CRISPR enzyme and guide RNA, once inside the nucleus of the cell, to find, through billions of letters, the matching DNA sequence. The first generation of the technology worked like a pair of molecular scissors. Once finding that target, it would cut the DNA, thereby shutting the gene off. Now, newer versions of the technology work like a molecular scalpel, surgically repairing the problematic letters with great precision. This can involve doing chemistries on the DNA, writing in specific letters, even changing the regulation of the genes. But as these technologies have gotten more sophisticated, the molecules have gotten larger, and therefore make it even more difficult to deliver gene editing technologies to the cells and tissue types where editing is needed to stop or potentially reverse disease. So today, access to the cells and tissue types with gene editing remains one of the biggest challenges for deploying CRISPR across a number of diseases. Today, there are two general categories of gene delivery systems that can take those CRISPR molecules inside the body. The first gene delivery system is LNP, or lipid nanoparticle, which consists of billions of tiny fat bubbles that can carry genetic payload. LNP delivery is like a cargo ship. It carries big, flexible shipping containers large enough to fit the CRISPR components. But most of these ships typically dock at the same port, in the liver. The second gene delivery system is AAV, or adeno-associated virus. It's a non-pathogenic virus that can target specific cell types. AAV delivery is like a drone. It can carry a small package to a specific location. It's lightweight, efficient, and it can fly under the radar, which is important because it can avoid triggering significant immune responses. But these drones can only carry a small payload, which means that the CRISPR medicine that's encoded within this particle must be compact and fit within the strict size limitations. The original CRISPR technologies have shown success in LNPs, but have been simply too large to fit within AAV delivery. To solve this problem, my colleagues and I discovered and developed tiny CRISPR systems small enough to be packaged within these non-infectious viral particles. When we first started this work, the consensus in the field was that these tiny CRISPR systems were likely just artifacts and probably not functional. But with persistent effort, we were able to show that these novel ultra-compact CRISPR systems were not just functional, but were in fact robust gene editors. And so now that has unlocked a whole new opportunity for gene editing, being able to develop ultra-compact CRISPR systems alongside all of the components needed for precision gene editing and still fit within the confines of tissue-specific delivery modalities. Today, we can readily edit cells outside of our body or in the liver. In fact, the world's first FDA-approved CRISPR therapy involves extracting blood stem cells from the body, editing them with CRISPR in a laboratory, and then reinfusing those edited cells back into the body. This process is long and expensive. It also involves chemotherapy to condition the patient for retransplantation of those blood stem cells. But it offers a functional cure for patients living with sickle cell disease. As for the liver, there are a number of diseases that can be addressed, and in fact, a number of ongoing clinical trials to look at indications like heart disease, which is one of the world's leading killers, and inborn errors of metabolism, rare metabolic disorders caused by a specific mutation, like the ones we heard about in baby KJ. But the biggest challenge we face is how do we get these CRISPR molecules in organs outside of the liver, across the body, in tissues like brain, muscle, heart, lung, and many others, because in order to address the broad scope of genetic disease, we must be able to deliver these medicines to the tissues where we can address that particular indication. Despite all the incredible progress, there remains significant challenges to achieve the safety, efficacy, scalability, and accessibility of CRISPR medicines for the more than 5,000 known genetic diseases. Patients can't afford to wait and deserve a chance at a cure. In fact, all of the advances in AI have made it even faster to decode the biology and underlying genetics, making it an accelerated pace to develop next-generation CRISPR medicines. And imagine what the world might look like if curing a loved one living with debilitating genetic disease is as simple as pairing an ultra-compact CRISPR system with a delivery modality and a guide RNA that has been reprogrammed for that particular disease. We believe that these ultra-compact systems will set the stage for the next chapter of gene editing. With each step forward, we are moving closer to a world where curing disease is no longer just a hope, but is becoming a reality. We are witnessing the future of medicine, the ability to rewrite the story of health by editing the genes in our body on demand. Thank you.